FDA recognition for the ClinGen VCEPs. Became official last Wednesday or Thursday or Tuesday, a day last week. It became official. And so I put a screenshot here from a few of the tweets from both the FDA, ClinGen, and NHGRI, all of which have links to where you could click out to learn more about sort of what this means and some of the background about this whole process. Additionally, this bottom screenshot is from the ClinGen website, which also has a link to go learn more about sort of what this means and look at the overall decision-making process for this. Oh yeah, so it does that. One link will link out to what the actual FDA recognition database means, and then so ClinGen is the first, and so now only one, that is a recognized database, and if you click on this decision summary, it does sort of give you an interesting breakdown of, what actually did FDA look at from this whole process, what is of interest to them, what are the requirements for groups going forward, what are they going to expect, some of which we'll get into during today's call, but it's also interesting to look at if anybody is interested in that. So the recognition went to the ClinGen variants, but as far as how do you actually see this information, one of the places is within ClinVar, and I'll talk about the evidence repo as well, but the first place, again, being ClinVar, so you can see for the ClinGen VCEPs that have sort of gone through this process now, oops, wrong way, all of them on both the search result page within ClinVar has this new FDA-recognized database link for any of them that have gone through this process. Additionally, on any of the actual variants, again, up at the top of the page, we'll have this additional tag next to the expert panel process, and then additionally down sort of in that summary table of classifications, again, this tag for the FDA-recognized database. So it is important to sort of just briefly distinguish that not every expert panel in ClinVar has now this FDA tag or has gone through this process. So right now it is limited to the ClinGen variant curation expert panels. The ones that have been submitted to ClinVar right now are listed here. The CDH one isn't live yet, but should be very soon. So the non-ClinGen expert panels like Enigma, Insight, CFTR2 weren't currently part of this process. I mean, there is still, in the future they could choose to go through these efforts to also get this FDA distinction, but they are still able to be expert panels in ClinVar. This FDA tag is more of like an additional little badge of honor for these groups. So these aren't being, downgraded or anything. They're just sort of a extra distinction for being able to go through this process of having a more standard and rigid way in which they go about their assessments and sharing a certain level of evidence with the community, so there is some transparency. And that's why the ClinGen VCEPs were able to get this recognition. All right, I think that's, yes, all I had for background at this point. Laura, if you want to go into more of your requirements, then I can jump back to talk about the data sharing BCI processes.
That sounds great. Thanks, Stephen. I'm going to pass the ball to you, Laura.
Okay. Thanks, Stephen, for giving everybody that background and sharing that exciting news with everybody. Okay. So can everyone see my slides now?
Not yet.
Okay.
Looks like it's coming up now, Laura.
All right, is this the right screen, I'm hoping?
Yep, Biocurator, Proficiency, Training, Expectations.
Excellent. Okay. So Stephen mentioned this is exciting news from the FDA about their recognition of our ClinGen variant curation expert panels, or VCEPs for short. One of the things that we discussed with them was whether they would be willing to do a kind of a pan-ClinGen approval. So instead of having to approve each VCEP piecemeal, whether they would just approve our methods. And so thankfully, they have gone ahead and approved our variant curation processes and our organizational management. So this means that the FDA recognition applies to the current ClinGen and future approved variant curation expert panel variant classification. So essentially, they've approved our process, which means that going forward, all ClinGen approved VCEPs will be FDA recognized. So one of the things that they did mention was that they required some more comprehensive SOP-like guidance for the VCEPs. So after a fairly detailed process, which was led by UNC's Julianne O'Daniel and supported by a whole host of people from all of the various grants, too numerous to name, we have detailed all of their expectations for VCEP approval and compliance in a document called the VCEP protocol. So for anyone who's seen the VCEP toolkit, the protocol is taking its place. It's a more detailed document. It lives at this URL, and if you're interested, probably it would be a good idea for everybody who works for VCEPs to go ahead and just read over this document just to see what they're expecting, and then Stephen's going to kind of hit some of the highlights about the new requirements that Biocurator should be aware of. And then the Variant Curation SOP Committee, which has a lot of people from this group on it, is in the final stages of developing the Variant Curation SOP, which will guide the curation process and training of all future VCEP biocurators. So we're looking forward to having that in hand probably early next year. And this is going to kind of lay out the training and the process for all new and current variant curation expert panel biocurators. So I've broken this down into expectations for new biocurators and for all biocurators. So to start, for new biocurators, the specific VCEP will be responsible for coordinating, monitoring, and recording training and proficiency of new biocurators who have not previously been trained. So that means that probably most of the people on this call would not be required to go through these processes, but since we are going to have new variant curators coming into some of the groups, including some new volunteers, we just wanted to make you all aware of what's going to be going on. So the FDA has asked that we have an experienced ClinGen trained biocurator who will work as a biocurator trainer and mentor to assist them with the training process, which I'll talk a little bit about in a minute, and then to guide the new VCEP curator through training curations and reviewing the initial curations until they've demonstrated proficiency with the core competencies, which I'll also talk about in just a moment. And then there will need to be a record of all biocurators who are proficient for that VCEP. So this is actually something that needs to be documented for all biocurators, and so you can see this down below that we need to have a current list of proficient biocurators. It doesn't need to be made public, but it needs to be on hand if we receive a request from the FDA or anyone else. So this is something that we could potentially be audited for. So biocurators and coordinators should start to think about this for their current VCEPs. And then all biocurators are expected to join this working group, and it's twice monthly calls. So does anybody have any questions about that before I talk a little bit more about the training process?
Hey, Laura, who do you envision is this Biocurator mentor? I know it could be different for different groups, but is this like typically the coordinator, or do you envision maybe some groups having a head Biocurator that would be that mentor? Has there been any discussion about that?
Yeah, that's a good question, Christy. So I think it's going to depend on the group. So I think for each of the groups that is taking on new VCEP curators, whether they're domain biocurators or volunteer biocurators, there would need to be someone who is willing to keep track of these folks. So it could either be a coordinator like you who's trained in the gene curation framework, or it could be a senior ClinGen trained biocurator like Marina or Erin or Courtney or Jenny or any number of other senior biocurators. And that would also go for folks who are ClinGen trained domain biocurators. So for instance, Jessica Master would be someone who would fall under that purview as well. But they would be responsible for recording and determining the proficiency of the new biocurator. So it would have to be somebody who was willing to take responsibility for appropriately training that new biocurator in their domain processes and making sure that they adhere and understand to the ClinGen framework as well. So does anyone have any other questions or comments about that? Okay, so I'm just going to quickly talk about some of the biocurator training and the biocurator proficiency assessment. So these are slides that Lisa Kurtz, who is leading the Variant Curation SOP committee, has presented to the steering committee. So these are pretty high level, just to give you an idea of kind of where we're going with it. So based on the gene curation biocurator training that was led by Erin Riggs yesterday and was extremely successful from what we've heard, what we're going to have is a level 1 training, which would be for all variant curators, and it would be essentially a standardized training for variant curators across ClinGen, and then level 2 would be specific to that particular variant curation expert panel's domain. So it would be getting more into disease mechanisms and things like that. And so the biocurator proficiency assessment would be this defined process for level 1 and 2 that we could use to say, okay, well, this person has determined or has demonstrated proficiency, and then we could go ahead and just put them on the list. So level 1 training would be what we're envisioning as a webinar after the first initial training, and it would be the same for all of the biocurators, and it would review the new variant curation SOP that's close to being done and the Richards et al. ACMG AMP guidelines paper, and this would be performed by the biocurator work group, and then the VCI development team, which is headed up by Matt Wright, would provide VCI training. And at that point, the curators would sign an attestation of attendance, and then they would be added to this group and to the listserv. So this would be the level of training for everybody. And then everyone would go to their level 2 training, where they would be designated to their particular variant curation expert panel, and then this would be done by each specific VCEP on their specific guidelines. So at this point, the new curators would be paired with their established curator mentors, who would then document their training and proficiency, and then they would sign this attestation form that they were that they had received training in the 3 core proficiencies, which we have discussed as being evidence identification, so does the curator know where to look for the necessary information, how to interpret the data, and are they thorough in their search process and in their interpretation. The second core proficiency is in the actual curation, so does the curator know where to put it in the VCI, and are they putting it in the right place, and is it comprehensive? And then finally, an evidence assessment. So have they assessed that particular evidence properly, and have they properly interpreted that evidence in the scope of the ACMG criteria and the VCEP specific guidelines? So yeah, the first 2 would be done by the level 1 training, as I just mentioned, for everybody, and then the level 3, or the third core competency, would be done by the VCEP specifically. And then, so this kind of goes back to before the training. We are still developing it based on the gene curation training. And then once that is developed, the genomic variant work group, which is led, I think, by Heidi, Krista, and Sharon, will review and approve it. And then we will get going with the training process. And we're looking forward to doing that because we have a lot of volunteers who are very interested in doing variant curation. I think that's the last slide. So does anyone have any questions?
So I'm going to ask a question. So there will be disease-specific experts who will be overseeing these specific disease group or disease-specific variant curation?
So I think more importantly than disease-specific experts are going to be experts in the actual ClinGen process for performing variant curation. So we would just need to make sure that we had somebody who was training the curators who can ensure that they're following the process that's been recognized by the FDA. Does anyone have any other questions? Feel free to reach out to me afterward as well.
Laura, can you hear me?
Jesse, is that you?
Yeah, hi. So I was actually wondering about the core competencies. I'm actually in the process of reviewing the SOP document still to give feedback on it. Thank you. Sorry, it just went real quick. Evidence identification, evidence curation, evidence assessment. Okay, yeah. I'm feeling like a piece that might be missing in this whole thing is how to search the literature, and how, I think the document as it stands is really good at walking somebody through popping information into the VCI appropriately, but I worry that we're not doing enough to train people on how to find information on find papers that cover a variant. Is that something that and I feel like that would be helpful for this level 1 training versus having it be something people do individually in the expert, in the expert panel portion.
Okay, well, I think that is definitely part of the evidence identification, and that's great feedback about the SOP. So, yeah, I definitely think that would be part of the level 1 training, and we can kind of look at beefing that up in the SOP. So thanks for the feedback.
Thanks. So any other questions for Laura before we move back to Stephen? Well, thanks so much, Laura. That was terrific. I'm going to pass this back to Stephen, who is going to talk with us about the VCI relevant requirements from the FDA.
All right. Can you see my screen again?
Yep.
Okay. Perfect. So I was going to go through a little bit about what is actually being shared out of the VCI and where, because I think it's important for people who are using the VCI to know what things that they write and enter is going to be shared publicly, as well as going through a few changes that have come about in the last week to how you can enter data in the VCI that sort of impacts this downstream sharing of your criteria and explanations. So first, an important part of the whole FDA recognition of the ClinGen was not just for the actual variant assertions, but also the evidence that supports them. So this was a key part in working with FDA, is to making sure that it was not just the classifications that we were making publicly available, but all of the supporting data. And they felt that that was a very important part of this entire process and was a necessary thing for us to reach this recognition. So you can see from some of the prior ClinGen EP classifications that were in ClinVar, the overall evidence summary FDA felt was insufficient to show really what evidence went into it. This was typically, the expert panels at the time were just listing which ACMG criteria were used, but there wasn't sort of that link between which publications were used for each source, how did they actually complete each one of these criteria. And this was largely due to the time that we weren't using the BCI yet. These groups had already sort of started their curations before this process, so when it came time to submit to ClinVar, we didn't really have that structure to be able to pull together. So the now key part has been now going through and sort of thinking through what is the actual evidence that we want to take out of the BCI and both be putting into ClinVar, as well as the evidence repo, so that we can complete this requirement from the FDA to have the evidence available as well. So overall, sort of how the workflow works is there's an export of this data that comes out of the BCI. The ClinGen Data Exchange Group converts it, makes it all pretty and makes sense, and then it goes on to both ClinVar and the ClinGen Evidence Repository, which I'll show some screenshots of both. And additionally, these both have links to each other, hyperlinks between each other. So if you're on one, you can click to go over and look at the other. And I'll show a bit of what the difference in what's being shared between these two places are. So looking at what comes out of the BCI to ClinVar, this gray box at the top is all of the sort of big picture information about the variant that's going on to ClinVar. Information like, what is your clinical significance? Again, date last evaluated. What was your assertion method citation? What condition was this assertion with regards to? And then also, as far as evidence, what goes along is your evidence explanation, so your free text summary that's entered in the BCI at the end that explains overall why you classified the variant, what you did, and summarizes the evidence, as well as any PubMed ID or citations that were entered into the BCI. See, I've kind of mapped here where all these different parts appear within ClinVar that come out of this BCI export. So now comparing this to evidence repository, it still is this basic information that goes on to ClinVar, but it sort of goes to the next level of also being able to show structured evidence. So for each ACMG rule assessment, being able to show what the explanation was for that specific criteria, as well as showing exactly what PubMed IDs went into it in a more structured form. So this information is often in this free text summary, but we also felt it was important to have the structured evidence so that, let's say in the future, somebody wants to say, I want to be able to pull out all variants that came from the P10 expert panels where they applied PVS1. So going through ClinVar, that would be a little hard to do. You'd have to mine free text data, whereas within the evidence repo, since this all will be structured, you could do better queries like that of wanting to look specifically at when a certain criteria was used or not. So here's a screenshot of a variant or what's shared from the BCI into the ClinGen evidence repository. So it's a lot of, again, some of the same information as ClinVar as far as, an overall variant description, showing different expressions of the variant. It even has links, again, to go look at this variant in ClinVar as well, has the overall classification, and then which criteria were met, and sort of who was the expert panel. And then also, this free text summary that also was, again, coming from BCI and in ClinVar about sort of overall summarizing the evidence and why you classified the way it did. But this bottom part is sort of what differs from what's in ClinVar, where for each criterion, it has that explanation as far as why this criterion was met, and below it are all the different pieces of evidence that were used for this criterion. So I've kind of blown up this bottom section so you can look at more in detail. So we're showing what is the overall explanation as to why PS4 was met, and then if there were multiple PubMed IDs, each one of those, as well as the explanation for why I used that PubMed ID for this criteria. And so all this information then now, again, in a structured format, is going into the evidence repo. As far as where this all comes from, I added in a side-by-side of the same variant in the BCI, so you can see exactly what you enter in the BCI shows up where here. So again, sort of your overall explanation for why that criteria was met. This PS4, that appears here as sort of the big picture tied to PS4. And then I had all these different publications. For each one, I added in a free text summary as to why I used that publication. And then all of those evidence summaries appear next to the link to that PubMed ID here in the evidence repo. Is there any questions about this whole process of the sharing between what's in BCI and evidence repo and ClinVar? Now I can keep going. Okay. So the next thing I want to talk is a few changes in BCI that will help a bit more with this sharing between ClinVar, and the evidence repository. So first, sort of a minor point, if you've been within the BCI in the last week, you've noticed that anytime you start a new curation is this PHI disclaimer that asks users to not enter any sensitive or unique information that could be used to identify a variant. So whenever you start a new curation, it'll ask you to either agree or disagree with this disclaimer before starting the curation, just again, because we don't want there to be identifiable or PHI data entered into the BCI. Okay, but the bigger sort of changes to go through is this change where now each criteria will have its own explanation box. So currently, in the old layout, this change happened, I think, on Friday. So pre-Friday, the way that a section like this would look, where there is a grouping of criteria, so in this case, functional studies, you have the option of these two different criteria, PS3 or BS3 for functional studies, but there's only this one explanation box to enter in why you met or not met any of these criteria, as well as, you can enter all these PubMed IDs down here below, but then there's just this one explanation. And this can be a little bit confusing at the end, because if you look at the very end, your sort of summary of your met and not met criteria, for those groupings, what you enter in that box appears with all of those criteria. For example, in this case, we have this explanation as to why we applied the functional assay PS3, but because there's only one box, the same description appears under the benign functional studies criterion. Same with the population data. This is a common place where this occurs. In this case, PM2 was met, so we're saying it was absent, but because there's only that one box, what we're saying here also is appearing under BA1 and BS1, which again can look a little bit confusing in the end with this layout. So on Friday, the BCI changed to now where each criteria will have its own independent explanation box, so that we can get a little more granular as far as what do I want to specifically say about one criteria versus another. Additionally, when you are entering a publication that support either of these, it will ask you to, that you can associate the publication with some of the criteria above. So let's say in this case you had one functional assay that was showing a deleterious effect and maybe one functional assay that was showing benign effect. You could add both of those here, tie each one to whichever criteria code is applicable, and be able to offer independent explanations for each.
I have a question about that, actually.
Yes.
So say you're only applying BS3 and you have an evidence blurb, and then you also put in some type of evidence for PS3, but it's not applied. On that evidence repo page, is the PS3 also going to show up even though you didn't apply it and you put evidence next to it?
So yeah, so right now what goes to the evidence repo is any criteria that is met or not met. So if you were to write something here but leave it as not evaluated, that would not go onto the evidence repo.
Okay.
Yes.
Thank you.
So you could, if you wanted to say met and provide an explanation for PS3 and not met for BS3, both of those would then go on to the evidence repo. And I have a slide at the end a little bit about when to do met versus not evaluated, because that was there was inconsistencies between how groups are currently doing that. So what to do about older curations? So what they've done right now is if you had previously done an assessment and entered in the explanation into just that one box that was tied to a grouping of criteria, right now that whatever you entered will appear under both. So in this case, this was initially just put in the box that was tied to both of these, but since on the BCI side they can't be confident always which one to map it to, right now it will appear under both with this whole data conversion, as well as any publications that have been entered won't specifically say which criteria it was tied to because that wasn't an option ahead of time. So I think for right now, it's fine for previous ones to leave it this way. I wouldn't necessarily think groups need to go back and edit these. If you're going in and reassessing the variant anyway, cleaning this up would make sense. But at this point, I don't think it's going to be required or anything for groups to go back and redo any of these to clear off from non-applicable sections. Oh, and then the last note now about the criteria selection. we did, again, notice some inconsistencies within how different variant creation groups were using these criteria. And actually, the FDA got a little confused as well because they were saying, oh, well, you had all these criteria in the end that you put not evaluated. If you had evaluated them, maybe that would have impacted your classification. And we realized the not evaluated maybe is not the best name for what it is, because the way we think of not evaluated is, it's you didn't look at it, or it's not applicable, or there's no evidence for that at all. So we've thought about maybe changing this in the future to not applicable or something like that. But essentially what we're saying is, MET, most people, I think, understand when to be using MET. But on the not MET side, we really feel like this really would only be used where there was evidence available, you evaluated it, and you determined in the end that it did not meet the criterion. So we have some variant creation groups that went through and for every criteria in the BCI either put it as MET or not MET, which then kind of looks confusing to the ones who didn't do it that way. So we do, again, feel like not MET really should only be when you did actually evaluate that criterion and in the end felt like whatever evidence you were looking at was insufficient to be able to apply that ACMG criterion.
Hey, Stephen, in the past, I remember us talking about maybe having options for both not evaluated and not applicable to distinguish the difference between those two things. Is that still on the table or no?
So yes, I think it's still on the table. We're trying to figure out, because we don't want to then, a few months down the road again, change it again to make sure that is sort of the final things we want, is like a MET, not evaluated, not applicable. Because it would be nice, I think, if we're rolling out not applicable, to have that sometimes be a automated answer. Like if I applied PVS1 for a loss of function, it'd be great if the criteria codes that are specific to missense variants automatically went to not applicable because they are not applicable if I'm looking at a loss of function variant. So I think being able to build in some logic like that would help with sort of having all these different options. But yes, sorry, in general, we are still considering adding not applicable as another option. All right, I think that is all I had. Any questions about BCI use, submitting data to ClinVar, or any questions about that process?
Hey, Stephen. So is the FDA now okay having not evaluated codes? Because I think we changed ours at the last minute, I think, to either MET or not MET because we heard there was concerns about them seeing not evaluated and thinking, just as you said, why didn't you look at those?
Yeah, so pretty sure at the end they did agree with us that actually, yes, because Julianne helped write these explanations. So I think at the end we did sort of tell them that not evaluated has a broader meaning than it looks by just the title. That it is also for cases where it just wasn't applicable or was no evidence. And again, anything that you mark as not evaluated doesn't go on to the evidence repo, but things that you mark as not met do go on. So it's also sort of a distinction is like there's times where you really do want to say not met, and if you're using it more for all criteria, you lose that granularity of times when you want to say, yes, I did have this evidence, but it just was insufficient to be able to use it.
Yep, that makes sense, and I agree. I think we really need a not applicable option sooner than later. Great.
Yeah, I will bring that. We have a call to the BCI group today, so I will ask them about that. If that's, I believe there is a ticket for that request, but we can check and see where it is in development.
Awesome, thanks.
Stephen, it's Jenny. I had a question about the development of standardized text for some of these free text fields. Is there any discussion about doing that, or is that something that should just be developed within each individual VCEP?
So yes, that is a good question. That is something we've definitely talked about, and I forgot to ever add that into here, is that another requirement for this whole process is that the groups do write that evidence summary at the end of doing their assessments. And we have had some back and forth on should there be templates for different situations? Should it sort of take whatever you wrote for each code and kind of put that together into a paragraph? Unfortunately, I think it's still going to be a little while before anything is settled or even implemented. But that actually maybe could be a discussion for a future Biocurator call, or inviting multiple people to join the BCI to see, how often we would want a standard template for all groups that use a specific code versus how often do the groups need something specific for their VCEP when they use that code. And sort of thinking through all those different options, because we also don't want to have it automatically write something that is incorrect and always needs to be edited or things like that. But we have some groups that started Google Docs, where they've been sort of putting their templates for each one. I think that is sort of the initial phase in we could do of this, of at least having, even if it's not within the VCI, having an open Google Doc where we can be sort of tracking a general ClinGen template for each criterion.
Great. Yeah, sounds like a good idea. Okay, any other questions for Stephen? All right, great. Thank you so much, Stephen. So I'm going to take the ball from you and talk a little bit about our variant curation SOP. Can everyone see my screen?
Yep.
Yep, great. All right, so just as a disclaimer, I have actually not been involved in development of the variant curation SOP, but I'm sharing slides that were made by Lisa Kurtz at UNC. She presented information on the variant curation SOP at our recent steering committee meeting, but Lisa was unable to join the call today, so I'm presenting on behalf of her and then the other folks who are listed here who have all contributed tremendously to development of this document. So thank you so much to this group for putting all this work into development of the SOP. So as Laura mentioned in her presentation, development of this SOP is really critical to training new and existing biocurators who are going to be involved in the variant curation expert panel process, and it's definitely something that was required for the FDA in order to have this standardized training process. So just to review, we have, of course, several things in place in terms of training biocurators. One is this call where you are all attending today, so thank you so much for being here. And that's going to be one of the requirements for new curators as well, is to join this group in order to be kept up to date with any resources or any procedures that we all need to know about that will help with the curation process. And then there's also VCI training as well, which is part of that initial training process. So any of the Stanford group can help with that, or any trained variant curators. And then there's also the demo version of the VCI, which can help with that initial training too. But what we're going to be focusing on today is this variant curation SOP. I'm going to try and go into presenter mode so that we can see this better. There we go, that's better. All right, so the overarching goal of the SOP is to guide variant curators through the process of curating and scoring evidence for the ACMG AMP assertion criteria. So all of the different pieces of evidence that we collect, so such as population data, computational and predictive analyses, functional criteria, and allelic and co-segregation data. And the SOP is really geared around the variant curation interface. So it really helps curators assess the evidence and also understand where to put it. So it really serves as a standardized training document for all ClinGen curators to perform variant curation, and then also forms a foundation for community curation volunteer training. So this is another new initiative that's really just starting out, is to get more engagement from the community in helping to curate these thousands of variants that are out there. So this SOP will be used by curation core curators, community curation volunteers, and also our expert panel or domain curators as well, and potentially could be used by others. As we know that the VCI is open to the public, so certainly any other folks who want to be using the VCI for variant curation could use this SOP. So the SOP, there's a good draft out there, and as Laura had mentioned, hopefully will be available for general use at the beginning of next year. So the SOP, it incorporates the ACMG standards and guidelines for sequence variant interpretation, of course, based on the Richards et al. Paper from 2015. It also incorporates recommendations from the sequence variant interpretation working group. So these are the general recommendations that can be used by across different groups for different criteria. For example, the PBS1 criteria that were published recently. Also give specific references to different expert panels, really as examples of a way to use these guidelines. And then it also incorporates information on how best to use the VCI. Where should this information go? So this is basically what it looks like. So a lot of different sections, obviously, and it has been set up based on the setup of the VCI. So for each tab in the VCI, there is a section in the SOP that helps to explain what information should go there, as shown here. So as I mentioned, the layout includes, for each section, includes the background, so the data type that's collected, where it can be found, and how to interpret it. If there are any limitations that should be considered, either of the data itself or of the VCI at this time. And then any criteria evaluations, so specific ACMG guidelines and specific instructions for use, and then any general considerations. So that is the layout for each section of the SOP. And so this is just showing some different sections here. So Lisa has included screenshots of the types of information that is available for these different tabs. And the group has tried to make it as user-friendly as possible, so including figures and tables to help explain some of the information where necessary. Also having dynamic links to external information sources, including ClinGen resources and VCEP pages. As I mentioned, giving examples where appropriate to help to explain some of the information, also providing appendices and resource sections. So this is just showing some of the different information that is in the VCI, so or in the SOP rather. So here are links to different useful websites for curators. And of course, as information is constantly changing with new recommendations coming out, maybe new SVI guidance, this will be a fluid document that will be updated as the resources change. So information then can be incorporated into the SOP and disseminated through this group and also the education working group as well. The document will be versioned, of course, so dates of the different versions, and that's important to keep into account as well. And this document will be available on the ClinGen website. So just like the gene curation SOP is available on the website, and as the gene curation SOP is updated, we always update the website and then get that information out to everyone through the Biocurator working group and other links as well. So the goal is that the SOP would be housed under the variant pathogenicity tab on the website as shown here. So final slide, just next steps. As I mentioned, there's a good draft out there. I know that Jessie mentioned that she's already been taking a look at the SOP, so thank you so much to Jessie, and thanks for that great feedback. So the next goal will be for the SVI and the Genomic Variant Working Group to approve the content of the SOP, and then hopefully it should be available early in the new year. And of course, we will let everybody know when the SOP is available and then disseminate that accordingly. So any questions about the SOP? As I mentioned, disclaimer, I have not been involved in this process. I know Stephen and Laura both have been. So any questions or anything we can help with regarding the SOP? All right. Okay, so I think, is Laura still on the call?
Yeah, hi, I'm still here.
Okay, great. You had dropped off as a presenter, so I'm going to add you Oh, that's funny. Let me make you a panelist again.
It crashed, and then when I signed back on, I don't know if you can still pass me the ball.
I can't make you a panelist for some reason. I'm not sure why, because I see you on here, but it's not letting me do that.
Okay, so I actually emailed you my slides, anticipating that
sure.
might happen.
Okay, let me, I see them. Thanks so much. I will bring those up. So just while I'm doing this, does anyone have any questions or anything that's not clear at this point? And of course, you're always welcome to email us after the call as well. Okay.
So while Jenny's bringing up the slides, and I know this has kind of been a dense call, so please feel free to stop me and ask questions. I'm going to talk about the community curation process and the group that's developing it. So as we all know, ClinGen is built on a culture of community engagement, as this group knows more than others, and knowledge sharing. And from the beginning, we've really envisioned crowdsourcing as a way to sustainably scale up curation. So apologies to people who may have seen this previously, but we're just trying to let everybody know really what's going on with the volunteer process, because I think maybe some people have heard about it but may have questions. So here's just a little snippet about what's going on. So we've really been thinking about crowdsourcing for a long time, but we didn't have necessarily the frameworks and interfaces in place to be able to support it. But now we do. So the Community Curation Committee, we're calling it the C3, really provides the necessary remaining infrastructure to organize for volunteer biocurators and their activities. So as some people may know, we are looking at two levels of community biocuration. So there's going to be one called baseline biocuration, which is going to use hypothesis to annotate evidence. The baseline biocurators are at will and are not required to participate in intensive training or join the expert panel or working group calls. Comprehensive biocurators, on the other hand, become expert panel members, and as such, they are the ones who are going to complete the rigorous training. They're the ones who are going to be joining the VCEPs who are looking for new volunteers, and they're the ones that we're really kind of envisioning the whole training process that I talked about, and attestation and assessment. They're the ones that process is really being set up for. So I'm going to talk mostly about these comprehensive biocurators and the process for that, since we still have some issues to work out with the implementation of the baseline biocuration. So there are 5 curation activities that are available for comprehensive biocuration. So gene and variant curation, actionability, dosage, and somatic cancer. And expert representatives from each of these 5 activities are members of this Community Curation Committee, as well as leaders and members of this group, the education work group, both of the biocuration cores that are associated with ClinGen, and also the data exchange and informatics groups. And so Liz Kearns from UNC is our coordinator, and I'd like to thank her for setting up some of these figures and numbers that I'm about to present. So this next slide is a 4-step process for all volunteers to complete a survey to answer some background questions. So we ask them about their stated preferences, and then we use those to assign them to a curation activity and to a particular working group or expert panel. And then once they're assigned, their appropriate representative, generally a coordinator, would contact them to schedule their level 1 training. And then after training, they go to their variant curation or expert or gene curation or whichever expert panel or whatever curation activity they're going to start with to complete their specific training and start curating. Next slide, please. So the figure on the right shows the number of people in each of the different professional roles that are volunteering to become comprehensive biocurators, which are shown in blue, and baseline biocurators, which is shown in red. So as you can see, of the 83 people who've taken the survey since it went live about 3 months ago, 85% are interested in becoming comprehensive biocurators. So this is not too terribly surprising given that we have been targeting genetics and genomics professionals to begin with. But interesting to see the different numbers of people in each of these different professional roles. So the first step for all ClinGen volunteers, whether they're referred by someone on this call or hear about us at a meeting or just are entirely new to ClinGen and find us via our website, is to complete a standardized survey, which is found on the ClinGen homepage and is also up on the C3 webpage. The survey has about 10 questions that help us gather information about the volunteers' interests, experience, their professional expertise, and then also their preferences for curation activity and working group or expert panel. And it just really helps us to place volunteers in their preferred groups, and also will help the different grants to inform their report writing, future recruitment of new volunteers, and potentially even help to inform the development of new expert panels in the future if there's a cadre of volunteer curators out there that could really help to jumpstart a new group. So this pie chart that is up on the slide now shows the number of people who indicated each of the activities as their first or sole choice. So surprisingly, a lot of volunteers have indicated an interest in more than one curation activity. In fact, we know that 63 out of 83 people who have already completed the survey said they would participate in an alternate curation activity if their first wasn't available. And so since obviously variant curation is still developing their training materials, we asked members then to rank their curation activity preference. So each of the 5 curation activity leaders contacted the volunteers who indicated that their curation activity was their first choice, and then simultaneously volunteers were also assigned based on their preferences to a particular working group or expert panel, though we did not tell them which one they were, we will not be telling them which one they were assigned to until after they have completed and attested to their training. Next slide, please. And so, the dosage gene and somatic cancer work groups have materials and have already scheduled their first training session. In fact, as I mentioned earlier, Erin scheduled 20 volunteers for a training session in gene curation yesterday and ended up with 63. And apparently had a really great and productive call. So I think a lot of folks showed up for a refresher, which was great. And then we also have some new gene curation expert panels, and I think some of those new curators showed up. So that was really fantastic. And then Shruti Rao and Deb Ritter have also scheduled 10 volunteers for training in somatic cancer curation early next year. And then, as we already talked about, variant curation is anticipated to begin in spring 2019. So Lisa has informed their volunteers about our timeline and also given them the option of temporarily moving to their next highest ranked activity. And then once they're trained, variant and gene curators will be contacted by the coordinator, and then they'll begin the training process that I already described. And then next slide. Okay, and then I think this more or less gets into what I had discussed earlier, but I guess just a couple of interesting things. The progress in baseline biocuration, the UNC Biocuration Core is piloting the tiered process, and that's yielding some really great results. And UNC developers are expected to begin working on an annotation tracker in early 2019. And then looking ahead to the ACMG 2019 meeting, we're planning to host a community curation workshop where we're going to briefly describe the process to attendees, and then have some of the expert panels who are looking for new volunteers kind of pitch their ideas to attendees. So we're thinking it's going to be kind of fun and engaging. We'll follow up with more details on this. And as you can see from the figure on this slide, we've gotten quite a few volunteers from ACMG. And so I think others have also been referred by colleagues and friends. So I guess keep up the effort, everybody, since we have lots of work still to go, but we seem to have a lot of interest. So thank you all for your effort as well. We certainly do appreciate each and every one of you. Does anyone have any questions before we go? I think we're a minute past the hour.
Thanks so much, Laura. That's really helpful.
All right. Well, feel free to email me if you have any